Hello everyone, Biochemistry channel is here with the topic titled Lipids-II. Don't forget to like the video before you start watching it. I wish it to be useful. Let's start to Lipids Part 2. The contents of this topic are glycerin-carrying lipids. And under this title, we'll talk about the glycerin, which is the backbone of these lipids. And then we will look to neutral lipids, which will focus on mainly triglycerides, which are the storage lipids. And the big family here is the glycerophospholipids, the lipids carrying a glycerin backbone and the phosphate group. And they share a common feature. They are the membrane lipids. Let's start our topic, glycerin carrying lipids. And here in the section header, you see a triglyceride, an example of a natural mixed triglyceride containing three different fatty acid residues. And here the blue one is a saturated fatty acid, the green one is a monounsaturated fatty acid, and the red one is our one of our PUFAs, the polyunsaturated fatty acids. Glycerin. It is also known as glycerol. It's a sweet, viscous, colorless, odorless, and liquid trivalent polyalcohol. And glycerol or glycerin, it forms the backbone of glycerolipids or the lipids carrying glycerin. It is miscible with water and ethyl alcohol in any ratio. It doesn't dissolve in ether and chloroform and benzol. If oxidized with hydrogen peroxide under the catalytic effect of iron salts in a slightly alkaline environment, a mixture of glyceraldehyde and dihydroxyacetone is formed. And do you remember what are these molecules? Glyceraldehyde and dihydroxyacetone. They are the simplest monosaccharides. So this means glycerin or glycerol can be used to get these two monosaccharides or vice versa, glyceraldehyde and dihydroxyacetone used to get the glycerin. And it happens in metabolism. Due to its water attracting and moisturizing properties, it is a sought after a substance in cosmetics and pharmaceuticals. Here you see glycerol, the hydroxyacetone, and the glyceraldehyde. And I would like to draw your attention to the fact that these three molecules are similar in terms of carbon skeleton. Look at them. They all carry three carbon skeleton or three carbon backbone. And this is extremely important from a metabolic point of view. They can transform into each other through both glycolysis and glyconeogenesis. And of course, these processes take place through enzymatic reactions. The processes are regulated by physiological conditions, which means regulated by hormones, stimulants, etc., and are based on phosphate derivatives. This means they have phosphate derivatives in metabolism, such as glyceraldehyde triphosphate, dihydroxyacetone phosphate, and also there are glycerol phosphates, and especially phosphate attached to the third carbon of glycerol. And these are the numbers of the carbons according to the UPAC rules, systematic rules. And for the glycerol also SN1, SN2, and SN3. And also when we check whether they carry an asymmetric carbon atom or not, as you see, glycerol doesn't carry any asymmetric carbon atom. So it's a chiral. But in real, it's a prochiral, or we can say it's a molecule which has a which there is a possibility to be a chiral after adding groups, and it happens. Such as when we add a phosphate group, the third carbon here, then the second carbon become a chiral carbon or an asymmetric carbon. Yes, after an information about the glycerol, now we are ready to add groups to the glycerol to get the glycerol lipids, and the first one is neutral lipids. Neutral lipids are esters of fatty acids with glycerol. They are the most common class of lipids. They are present in all animal tissues in greater or lesser amounts. Neutral lipids are storage lipids and have three important functions. They are reserve nutrients. They act as insulators against heat loss from the body. They act as a cushion to protect internal organs against the outside. There are two major advantages to using the natural lipids, and I mean the triglycerides, a member of the biggest member of the natural lipids. And triglycerides are fuel molecules. They are stored as a fuel instead of polysaccharide, such as glycogen and starch. And so we have the glycogen we discussed in carbohydrates, which is a storage for the glucose, and when needed, it's essential. An alternate fuel is triglyceride. So the organism can also store triglyceride as a fuel. And we know the triglycerides can yield more energy than a carbohydrate, than the glucose. It's about the fatty acids they carry. So the carbon atoms of fatty acids are more reduced than those of sugars, and the oxidation of triglycerides yields more than twice as much energy per gram compared to the oxidation of carbohydrates. Triglycerides are hydrophobic molecules, and therefore they store as dehydrated. To store glycogen, we need water. The glycogen is highly hydrated molecule. So the organism carrying fat as fuel does not have to carry the extra weight of hydration, which means the water associated with the fatty acids. Actually, there's no association, but in polysaccharides, water associated the polysaccharides. In humans, adipose tissue, composed mainly of adipocytes, as a cell type of adipose tissue, is found under the skin, in the abdominal cavity, and in the mammary glands. And moderately obese people who accumulate 15 to 20 kilograms of triglycerides in their adipocytes can meet their energy needs for months by utilizing their fat stores. In contrast, the human body can store less than a day's energy needs in the form of glycogen. But still, it's so important, especially when an animal or a human uh, needs to give a rapid response to any change in its surrounding. And carbohydrates such as glucose offer some advantages as fast metabolic energy sources, one of which is their ready solubility in water. But the triglycerides, actually the fatty acids in their structure, needs more time to convert them to energy. In some animals, triglycerides stored under the skin serve not only as energy stores, but also as insulation against low temperatures. Seals, penguins, and other warm-blooded polar animals or animals living in the polar regions or in the cold regions of the world are abundant in triglycerides. Also, in hibernating animals such as bears, the large fat reserves that accumulate before hibernation serve the dual purposes of insulation and energy storage. Let's look at the classification of natural lipids. We have the mono D and triglycerides, the main title here. And mono D occur during the metabolism, either in the synthesis of triglyceride or the breakdown of glycerides. And there are two origin. We have two origin: fats and oils, animal fats or triglyceride in animals, and oils or herbal or vegetable oils. And we have another two class which I only define them in this topic: glycerin ethers and the glycosylglycerins. Let's start with the mono, D, and triglycerides. And the three here means three. D, two, mono, one. Remember the UPAC multipliers. And also remember that the glycerol carry three alcoholic groups, which means possible three regions that can undergo esterification. If one alcohol group of glycerin is esterified with one molecule of fatty acid, monoglyceride is formed. If two alcohol groups esterified with two fatty acids, triglyceride is formed. If all three alcohol groups of glycerin are esterified with three fatty acids, triglyceride is formed. In general, the structure of fats is in the form of triglycerides. We know the fatty acids, we don't want all fatty acids to be in a free form because they are acids, so they change the pH of the environment. And the term triglyceride is often used for fats and also oils. The triglycerides also there are other names or the synonyms which are the triacyl glycerol or triacyl glycerin or triacyl glyceride. But I prefer to use triglycerides. In most eukaryotic cells, triglycerides form a distinct phase of macroscopic fatty droplets in the aqueous cytosol and serve as metabolic fuel stores. In vertebrates, specialized cells called adipocytes, especially in adipose cell or the fat cells, store large amounts of triglycerides as fat droplets that nearly fill the cell. And here you see the illustration or the diagram of the triglyceride, which composed of one molecule of glycerol and three molecules of fatty acid. And all fatty acids can be identical or different, or maybe two identical, the third one is different. And I said the triglyceride occurs with an esterification reaction, and alcohol over glycerol is esterified with the fatty acid, the carboxyl acid of fatty acid. And in general, if you remember from organic chemistry, we say the esterification is a reaction type which occurs between alcohols and acids. And here the alcohol is glycerol, acid is fatty acid. And during this reaction, 1 mole of water is removed. And this is also a condensation reaction. And this reaction gives an ester group here. This is the ester group. And this is a triglyceride example here, another example, which we can easily identify the glycerol backbone here. And these are the fatty acids. And we can give the name of these fatty acids because we did it in the lipids part one. And now it's easy to say the fatty acid attached to the first carbon, SN1, is a saturated one, and in the two and third are unsaturated one, and even the third one is a polyunsaturated fatty acid. And you can try to name the fatty acids, both systematic naming and the UPAC names, and also for the unsaturated ones, you can do the omega classification. And the one is the palmitic acid, Second one is oleic acid, omega 9 and omega 3, the essential alpha linolenic acid. So this means there are too many different kind of triglycerides or triacyl glycerols or shortly TGs in nature. And here, as a concept, as a conceptual approach, we just focus on the triglyceride, the general term. We do not enter the deep points, or we don't discuss or talk about the different kind of triglyceride. Just learn the concept. And this is a name here. So this means depends on the fatty acid carried by the triglyceride. So we can give the name such as 1-palmethyl, 2-oleoyl, and 3-alpha linolenoyl glycerol, or shortly POL, palmitic oleic linolenic acid. And as I said in the previous slide, the fatty acids in triglycerides can be identical or different. And if all esterified fatty acids are the same, we call it the simple or homogeneous glycerides. And the glyceride term also used to represent the triglyceride. Both animal fats and vegetable oils are highly complex esters of fatty acids with glycerin. This is why we call glycerides a short, but I prefer always to say triglyceride to put a distinct definition or a definite and a concise definition. And for example, if 3 moles of stearic acid are bound to glycerol, it is called stearine or tristearine. If we have a triglyceride carrying 3 oleic acid, we call it olein or triolein. If the fatty acids esterified glycerol to form glycerides are different from each other, and even the one is different, it's okay to be a mixed or heterogeneous glyceride. And the example we did before is a was an example of heterogeneous glycerides. Yes, the physical properties of triglycerides. The fatty acids in the triglycerides may be saturated and or unsaturated. Animal fats and vegetable oils are triglycerides with different physical properties. Animal fats have a high melting point. They are solid at room temperature. Vegetable oils have a low melting point. They are liquid at room temperature. To determine whether a triglyceride is animal fat or vegetable oil, we should look to the fatty acids they carry. Remember the physical properties of the fatty acids. We talk about the unsaturated ones in the cis formation, which are the common ones in nature, has have lower melting points than the ones saturated. And so this also affect the physical properties of triglycerides. So the animal fats, solid fats, have saturated fatty acids in relatively high proportions and are generally of animal origin. And without double bonds, the three side chains of saturated lipid extend parallel to each other and have a high melting point. And pork oil, butter, whale shell are examples for the solid fats or the examples which carrying high proportion of saturated fatty acids or triglycerides. Liquid oils or vegetable oils have a higher percentage of unsaturated fatty acids and are usually of vegetable origin. Unsaturated lipids, which are mainly triglyceride carrying unsaturated fatty acids, even 1 mole in a cis form, in the cis double bond, forms a band in the side chain and makes it more difficult to pack efficiently in solid form because it will decrease the space occupied by the molecule, thus decreasing the melting point. So they have lower melting point. And the vegetable ones, corn oil, soybean oil, or sunflower oil, they contain unsaturated fatty acids. Actually, they contain triglycerides carrying unsaturated fatty acids in their structure. And here's two examples of triglycerides. And look at here, the triglyceride. And as you guess, the red balls represent the oxygen. So this is the carbon backbone of fatty acid here and also here. But look at the shapes. This is a straight chain, a linear structure, but here there are bands. There are bands which has a more which has a disorganized structure. But here there is an organized structure, and this is the space filling models of two different triglycerides. And let's check which is a possible animal origin and which is vegetable. And this is the animal fat example, which is highly saturated, and all are saturated, as you see here, because there is no any band even in one of them. And in vegetable, all fatty acids has a band here, as you see, band, which is possible. The origin is possibly from a vegetable. And saturated fatty acid chains pack together well in triglyceride molecules as well as they do in single fatty acid molecules, as seen here. Furthermore, saturated fat molecules also pack together well. Saturated fats are solids at room temperature. And triglycerides composed of unsaturated fatty acids do not pack together well. So unsaturated fats are generally oils at room temperature. And the fats is often used to refer to solid, mostly saturated lipids, and the terms oil is often used to refer to the liquid lipids or unsaturated fats. And this is also important for the cell membranes because the cell membranes also carry fatty acids, and that kind of fatty acids, which I mean the unsaturated fatty acids, is important for the integrity and for the fluid character, and also this means the survival of the cell and its function. The melting point of triglycerides is higher than the melting point of the fatty acid they carry. Although short chain fatty acids are clearly soluble in water, long chain fatty acids are insoluble and soluble in organic solvents. The specific gravity of fats is lower than that of water, and the pure glycerides are colorless, odorless, and tasteless. The yellow color of butter is due to the presence of vegetable pigments called carotenes and xanthophylls. Unsaturated fatty acids of liquid oils can be solidified by saturating with hydrogen, and these structures solidified by hydrogen are called margarine. And here was an example of a triglyceride. As you see, all fatty acid in the backbone here, all are saturated. There's no any double bond in the carbon chain of fatty acids. And here is the some food sources which are rich in that kind of triglycerides: meat, cheese, butter, and they are solid at room temperature. And there is also a relationship between the blood cholesterol level and the triglycerides. But this is a very simple approach. There should be a question mark here. We should actually discuss more about this uh, this definition or this mention here. But this is not the topic for lipids part two. And another type of triglyceride, as you see here, one of the fatty acids in the structure is unsaturated, even polyunsaturated fatty acid. As you see, there are two double bonds in the cis formation. So one or more double bonds in the carbon chain make the triglyceride liquid in room temperature. It doesn't necessary for the all fatty acids to be unsaturated. Just one is enough because it will cause bonding, thus the disorganization or not to pack the molecules in a compact shape. And here were some foods which are rich in that kind of fatty acids, such and vegetable oils, nuts, soybeans, and the fish also salmon, mackerel. And here also the avocado, which is very famous nowadays. And they are liquid at room temperature. They decrease the blood cholesterol level. Actually, change the definition. They are good in terms of health, we can say, but we need more discuss about this. And you can check from the web, especially from the scientific indexes such as PubMed, Web of Science, or Scopus. And also you can try to look to Google Scholar. And look at here, we have another triglyceride, and as you see, the one of the fatty acids carry double bonds, but it's a trans double bond, which make the fatty acid here behave like a saturated fatty acid. There is no band at the point of double bond due to the trans nature, which make the triglycerides semi-solid at room temperature. And these are the food sources which are rich in that kind of triglycerides. Let's talk about the chemical properties of triglycerides. And the one is hydrolysis. Triglycerides are hydrolyzed by boiling with acids or bases under normal pressure, or with water under high pressure, or by catalytic action of certain enzymes such as lipids, and especially the pancreatic lipase. Pancreatic lipase. Because there are too many lipases in the body, but here, especially for the digestion, we mean the pancreatic lipase. But also there are phosphoprotein lipase, liver lipase, kidney lipase, etc. But the one which is responsible for the digestion of lipids, fats, oils in the small intestine is the pancreatic lipase. And it's an hydrolytic enzyme. And the hydrolysis means breaking the ester bond by adding water. Remember the formation of triglycerides? I said it's an esterification reaction, and during esterification, one mole of water is removed from the bond. And if we add the one mole of water, we break the bond. And we need an enzyme, lipase. And the formation of unpleasant odor and taste in oils known as aggravation or rancidification is partly due to the release of fatty acids. And hydrolysis of triglycerides, in a complete hydrolysis, the products will be 1 mole of glycerol or glycerin and 3 moles of fatty acids. And look at that molecule. An example, we have a tristearine triglyceride carrying 3 moles of stearic acid. And the three bonds here are drawn in red, which will be broken by adding water in hydrolysis and using an acid catalyst here. And the result will be glycerol and 3 moles of stearic acid. This means hydrolysis breaks the free single bonds, free ester bonds between the carbonyl carbons and the oxygen atoms of the triglyceride or the glycerides or the esters. Since tristearine contains three identical R groups, which are the stearic acid on the carbon-carbon, three molecules of the fatty acid stearic acid are formed. If the glycerol carrying different kind of fatty acids, at the end we have different kind of free fatty acids: stearic acid, palmitic, palmitoleic acid. And the two are these are the saturated. This is an unsaturated fatty acid. Saponification. A soap is prepared by the basic hydrolysis of a triglycerol. Heating an animal fat or vegetable oil with aqueous base hydrolyzes three esters to form glycerol and sodium sulfate 3 fatty acid. And we have a saponification value. It is expressed by potassium hydroxide in milligram required to saponify 1 gram of fat. And the saponification value is regarded as an index showing the average molecular weight of fatty acid. And some soaps, especially sodium and potassium soaps, are easily soluble in water because the dissolution of the carboxylic groups is quicker. And here is an example of sodium stearate having a nonpolar tail and an ionic polar head. And sodium stearate is the sodium sulfate stearic acid and 18 carbon saturated fatty acid. And here is an example of sodium oleate, a soap obtained from vegetable oil, olive oil. And olive oil is rich in triolein, a homogeneous triglyceride, carrying 3 moles of oleic acid. And its soap is sodium oleate. Separation of fatty acids. It is the separation of fatty acids in an oil by detecting them. With that reaction, it is possible to separate the fatty acids in an oil in free form. So we can obtain free fatty acids. And hydrogenation. It is the saturation of unsaturated bonds in fats with hydrogen. And unsaturated bonds of fatty acids in oils are saturated with hydrogen and margarines are obtained. Halogenation. The unsaturated bonds of fatty acids are saturated with halogens such as chlorine, bromine, iodine. And we have an iodine volume. It is the mass of iodine in grams that is consumed by 100 grams of a fatty acid. And the iodine value is an indication of the relative unsaturation of oils. Oxidation. It occurs when ozone or the molecular oxygen is added to the double bonds of the unsaturated fatty acids. And the rancidification of the oil is partly due to the hydrolysis and the partial oxidation. Acetylation. Fats or oils which carry fatty acids with free hydroxyl groups are acetylated with acetic anhydride. And we have an acetal number here, a measure of free hydroxyl groups in fats or oils determined by the amount of potassium hydroxide used to neutralize the acetic acid formed by saponification of acetylated fat or oil. And the acetal number gives the average amount of oxyacids in the fat or oil. An oxyacid, oxoacid, or ternary acid is an acid containing oxygen. Specifically, it's a compound containing hydrogen, oxygen, and at least one other element, and with at least one hydrogen atom bonded to oxygen, which can decompose to produce the hydrogen cation, the proton, and the anion of the acid. Rancidification. It is the process which causes a substance to become rancid, that is, having a rank, unpleasant smell or taste. Acid value. It is the mass of potassium hydroxide in milligrams that is required to neutralize 1 gram of free fatty acid. It is regarded as an index of rancidity caused by free fatty acids. And we have another value, Reichert value or Reichert-Maisel-Volney value. It is equal to the number of milliliters of zero point 0.1 normal concentration hydroxide solution necessary for the neutralization of the water-soluble volatile fatty acids distilled and filtered from 5 grams of a given saponified fat. And the Reichert value is an indicator of how much volatile fatty acid can be extracted from fat through saponification. And rancidification or to become a rancid also means to become bitter. What are the causes of rancidification? It may result from the hydrolysis of glyceraldehydes in a fat to glycerin by free fatty acids or to mono and diglycerides by free fatty acids. It may result from various oxidation processes and also may result from beta oxidation of free saturated fatty acids. And lipid-rich foods can spoil and rancidize when exposed to oxygen in the air for too long. The unpleasant taste and odor associated with rancidity results from the oxidative cleavage of double bonds in unsaturated fatty acids, which produces aldehydes and carboxylic acids with shorter chain length and thus higher volatility. And these compounds easily pass from the air to your nose. And we finish the triglycerides, and let's look to just the definitions of the other natural lipids. The one is glycerin ethers. They are lipids in which one or more of the carbon atoms on glycerol is bonded to an alkyl chain with an FAO linkage, as opposed to the usual ester linkage. It's found in animal tissues. And glycosylglycerols or glycerins, a class of glycolipids structurally analogous to phospholipids. They are the major glycolipids of plants and microorganisms but are rare in animals, and they contain high levels of linoleic acid. And this is the end of natural lipids. It's time for glycerophospholipids. But before moving on to glycerophospholipids or phosphoglycerides, it is necessary to briefly explain the concept of phospholipid. Phospholipid is a more general expression and briefly refers to any lipid molecule with phosphoric acid residue. In other words, I'm talking about lipids carrying phosphate in their structure. And phospholipids are defined as compounds made by fatty acids and phosphate containing compounds with alcohols, and here the glycerol. And there are two types of phospholipids. And the one is glycerophospholipids or phosphoglyceride, which the lipids part 2 includes glycerophospholipids. They are the phospholipids carrying phosphoric acid esterified with glycerin. And the second one is sphingospholipids, and lipids part 3 include sphingospholipids. They are the lipids, they are the phospholipids carrying phosphoric acid esterified with sphingos in alcohol. So the point which they differ is the alcohol used as a backbone. In the phosphoglyceride, the backbone is provided by glycerin and sphingolipids, sphingospholipids. It is provided by sphingosine alcohol. Phospholipids are both structurally and functionally significant. Phospholipids are the basic building blocks of cell membranes. And remember the basic structure of a cell membrane, it carries a lipid bilayer structure, and that layers are provided by phospholipids. They are important components of lipoproteins, the carrier of lipids, and they function in intracellular signal transduction pathways like lipid signaling. They are involved in blood coagulation. They are found in the bile secretion, and they are involved in the structure of the pulmonary surfactants. And there are some examples in the parentheses that we will study all of them, such as platelet-activating factor, phosphatidylcholine, phosphatidylcholine, or phosphatidylserine. Yes, after this brief explanation, it's time to start glycerophospholipids. Glycerophospholipids, also called phosphoglycerides, are membrane lipids in which two fatty acids are attached to the first and second carbons of glycerol by ester bonds, and the highly polar or charged group is attached to the third carbon by a phosphodiester bond. Glycerophospholipids are called derivatives of the parent compound phosphatidic acid according to the polar alcohol in the head group. For example, phosphatidylcholine and phosphatidylethanolamine have choline and ethanolamine as polar head groups. And here is the pattern of a phosphoglyceride or a phosphatidylglycerol or glycerophospholipids, and it's the phosphoric acid and phosphodiester bond having two R's. And this is the pattern in the cells. It's donated hydrogen. Here's an ionic state. And the pattern is the glycerol, 2 moles of fatty acid, and at the top carbon of glycerol, a phosphate group is attached. And this attachment makes the second carbon chiral. This is why we call the glycerol is a prochiral molecule. And the second R group here attached to the phosphate group with ester bond. This is why here we call the phosphodiester bond. And R is typically one of two different groups, either choline or ethanolamine, or also serine occur in the all phosphoglycerides and also in sphingospholipids. But the R's here, the groups attached to the first carbon and second carbon, the common ones are the fatty acids, but also we observe the derivatives of fatty acids. And in all these compounds, in all glycerophospholipids, the hot group is attached to the glycerol, as I said, we have phosphodiester bond in which the phosphate group carries a negative charge at neutral pH, negative charge at neutral pH. And here we have an alcoholic group again. Actually, there is a carbon backbone carrying an alcoholic group. And the polar alcohol can be negatively charged, as in the phosphatidylinositols, can be neutral, as in the phosphatidylserine, or positively charged, as we observe in phosphatidylcholine and ethanolamine. And these charges contribute greatly to the surface properties of membranes. And the fatty acids in glycerophospholipids can be very diverse. And the distribution of molecular species is specific for different organisms, different tissues of the same organism, and different glycerophospholipids in the same cell or tissue. So highly diverse. And the first glycerophospholipid is phosphatidylcholine, also known as lecithin. And phosphatidylcholine is a glycerophospholipid derived from phosphatidic acid. It is characterized by the presence of choline in its structure. And the fatty acids in its structure are usually unsaturated and the other unsaturated. But don't forget, there is also two fatty, there can be also two saturated fatty acids in the structure of lecithin or phosphatidylcholine. And let's look at the pattern of a phosphatidylcholine. Also, I can say this is a type of formula. 1 mole of glycerol, glycerin-glycerol, 2 moles of fatty acid, 1 mole of phosphoric acid, and till here, this pattern is called the phosphatidic acid. This is why I said they are the derivatives of phosphatidic acid. And the molecule which makes our glycerophospholipid phosphatidylcholine is the choline. And choline is a nutrient found in many foods and the brain, nervous system, the muscle, and it requires choline for the activities, for some activities or some functions. So they are essential molecules and also considered as vitamin by some authors. Let's let's look at the more detail about the lecithin molecule. And as you see here, the two given fatty acids are saturated fatty acids. And this is the famous, well-known representation of a phospholipid carrying a polar head, a pin-like head, and the two tails. And here the tails represent the non-polar parts of the molecule, which are provided by the hydrocarbon chains of the fatty acids. And the polar head is provided by the phosphate group, and the polar group here is the choline. And lecithin actually can be saturated with both fatty acids, and one saturated and one unsaturated. An unsaturated one is located at the second position generally. And lecithin is a generic name or a generic term used to describe a class of fatty substances that are a mixture of phospholipids. And the term lecithin is derived from the Greek word lekitos, which means egg yolk, as lecithin was originally isolated from egg yolk. And the main components of lecithin are phospholipids, and the main one is the phosphatidylcholine. This is the major constituent of lecithin. But also there are others, such as inositol contain ones. But the unique property of lecithin is provided by the choline. And actually, these are some kind of formal names. But today, we mainly prefer to say phosphatidylcholine to be more concise that what is referring or which type of glycerophospholipid is referring. And the unique property of phospholipids, glycerophospholipids, is their amphoteric nature, meaning they have both hydrophilic, the polar heat provides the hydrophilic character, which means the water attracting, and a hydrophobic region, water repelling region, and non-polar tails. And this makes phosphatidylcholine and also other ones, this is similar feature, an excellent emulsifying agent helping to stabilize and disperse mixtures of water and oil. And remember, in the lipid layer of a cell membrane, polar head locates either outer surface of the cell or inner surface of the cell, which interact with the liquid outside or inside of the cell, which means cytoplasm and extracellular matrix. And nonpolar tails are stay away from the liquid regions. They do not dissolve in water, but they have a great affinity for water. This is perhaps why it is one of the most important substances of protoplasm and one of the important building blocks of biological membranes. And phosphatidylcholine is an important phospholipid found in cell membranes and is particularly abundant in the membranes of liver cells. It is also an important component of lipoproteins involved in the transport of fats in the blood. In its deficiency, the important disorders such as fatty liver and hemorrhages in the kidneys are observed. And the enzyme phospholipase A, phos- phosphorylase A, can partially hydrolyze lecithin. And this enzyme can be found in snake venom, wasp venom, and some microorganisms. And when hydrolyzed by the enzyme phosphorylase A, 1 mole of fatty acid, 1 mole of lecithin, or 1 mole of fatty acid is separated, which I mean the fatty acid as a component of lecithin. And the remainder is called the lysolecithin or the lysophosphatidylcholine. And this molecule has a strong hemolyzing effect. And the hemolyzes mean a rupture of cell membranes of the erythrocytes, the red blood cells. Another glycerophospholipid, phosphatidylethanolamine. Phosphatidylethanolamine, one of the cephalins, is a glycerophospholipid derived from phosphatidic acid. It is characterized by the presence of ethanolamine, and the other name is cholamine, in its structure. And ethanolamine is synthesized from the serine amino acid. The fatty acids in its structure are usually one saturated and the other unsaturated. And this is the pattern of, or a building pattern of phosphatidyl ethanolamine, 1 mole of glycerin, 2 moles of fatty acids, which one saturated, the other unsaturated, 1 mole of phosphoric acid, and which is a phosphatidic acid. This is why also it's a derivative of phosphatidic acid. And the molecule which makes it unique is ethanolamine or the cholamine. And cephalin, you look at, I said, it's one of the cephalins. Cephalin is an older term that was historically used to describe a group of phospholipids, including phosphatidylethanolamine and phosphatidylserine. And these phospholipids were initially isolated from brain tissue, hence the name is cephalin. And over time, as our understanding of phospholipids has advanced, the use of term cephalin has diminished, and these molecules are now more commonly referred to by their specific names, such as phosphatidylethanolamine and phosphatidylserine. This phospholipid is an important part of the lipid bilayer that forms the basis of cell membranes. The specific regulation of phospholipids in the membrane is crucial for maintaining its structure and function. And phosphatidylethanolamine is found in all living organisms, and its distribution can vary between different cell types and organelles within cells. It plays a role in membrane curvature and fusion and contributes to the overall fluidity and stability of cell membranes. In addition to its structural role, it is involved in several cellular processes, including lipid metabolism and autophagy. And autophagy means the cellular process responsible for the breakdown and recycling of cellular components. And phosphatidylethanolamine also acts as a precursor for other lipids. Yes, here you see the pattern or the molecular structure, basic structure of two glycerophospholipids, the common ones, phosphatidylcholine and phosphatidylethanolamine. And as you see, they are the phosphatidic acid derivatives. This part is the phosphatidic acids. The R here represent the hydrocarbon chain of 2 moles of fatty acids. And the choline make the molecule phosphatidylcholine, and ethanolamine makes it the phosphatidylethanolamine. And both are the most abundant glycerophospholipids in eukaryotes. And both are the two main phospholipids distributed asymmetrically in the plasma membrane. The majority of the phosphatidylcholine is localized in the outer layer of the membrane, whereas phosphatidylethanolamine is enriched in the inner layer of the cell membrane. Besides their role as major structural components of all cellular membranes, both fulfill various other functions. Phosphatidylcholine is an important source for the formation of second messengers and lipid mediators. Distribution of its production inhibits cell proliferation, differentiation, and membrane movement across the cell. On the other hand, phosphatidylethanolamine plays important roles in autophagy, cell division, and protein folding, and is a precursor for the synthesis of various protein modifications. Furthermore, both are intermediates in the synthesis of other class of glycerophospholipids, which will discuss the metabolism of these glycerophospholipids in the Biochemistry-II course. Yes, a new glycerophospholipid, phosphatidylserine, is another type of phospholipid, another type of glycerophospholipid, and the cephalin, and is an essential component of cell membranes like phosphatidylcholine and phosphatidylethanolamine. However, phosphatidylserine, and as in the name, is unique that it contains the amino acid serine as its polar head group. And look at the pattern. Again, it's a derivative of phosphatidic acid and contains 1 mole of serine. Phosphatidylserine is found primarily in the inner layer of the cell membrane facing the cytoplasm. It plays a crucial role in maintaining the integrity and fluidity of cell membranes. Phosphatidylserine is involved in various cellular processes including apoptosis, which is defined as the programmed cell death. And during apoptosis, phosphatidylserine is released on the outer surface of the cell membrane and signals immune cells to engulf and remove the dying cell. It also participates in coagulation, cell signaling, and regulation of membrane processes. And phosphatidylserine is involved in the coagulation cascade by providing a surface for the aggregation and activation of clotting factors. And this process is essential for the formation of blood clots, a protective mechanism to prevent excessive bleeding. And phosphatidylserine, as I said, is normally found on the inner layer of the cell membrane. However, when muscular injury occurs, platelets are activated and phosphatidylserine is exposed on the outer surface of the platelet membrane. And this acts as a signal for the initiation of the coagulation cascade. Phosphatidylserine has been studied for its potential cognitive benefits too. And plasmalogens. Some glycerophospholipids have ether-linked fatty acids, and the one is plasmalogens. Plasmalogens are a type of ether phospholipid containing a vinyl Fe bond at the SN1 position of the glycerol backbone instead of the ester bond found in most other phospholipids, as we see in the phosphatidylcholine, phosphatidylethanolamine, and phosphatidylserine. So the difference is here at the fatty acid attached to the SN1 position. It's attached with not the ester bond, but the ether bond. So a different pattern is seen. They are abundant in cell membranes, especially in brain and heart tissues. And vertebrate heart tissue is uniquely enriched in ether lipids. About half of the heart phospholipids are plasmalogens. Platelet-activating factor, shortly PUF, is an ether phospholipid like plasmalogens. It has an ethyl-linked long alkyl chain at the carbon 1 position and an ester-linked acetyl residue at the carbon 2 position. It is synthesized by basophil leukocytes and is a strong signaling molecule. It has various effects in many tissues. It stimulates platelet aggregation and release of serotonin, a vasoconstrictor from platelets. It also exerts various effects on liver, smooth muscle, heart, uterine, and lung tissues, and plays an important role in inflammation and allergic response. Yes, my dear friends, here you see plasmalogen, molecular pattern of plasmalogen and the platelet activating factor, the PUF. As ethyl lipids, they are the little lipids, where the most glycerophospholipids have an ester-linked fatty acids. These two ethyl lipids have an ethyl-linked fatty acid. But what is the difference? Focus on the yellow rectangle here, showing the ethyl link or ether bond. In plasmalogen, it's a vinyl ether, ethyl link alkene or alkenyl chain. Look at here, there's a double bond. But in activating factor, it's an ethyl link alkane or alkyl chain. There is no double bond. And the other major difference between plasmalogen and platelet activating factor, there is a fatty acid attached to second carbon in the plasmalogen. But in platelet activating factor, there is an acetyl group attached to the second carbon. And what about the polar heads? The plasmalogen carrying mainly ethanolamine, it may also be choline in plasmalogens, but in platelet activating factor, it is choline. But in plasmalogens, it may be choline instead of ethanolamine. But platelet activating choline, platelet activating factor, the head group is choline. The phosphatidylglycerol, also known as cardiolipin, consists of two molecules of phosphatidic acid linked together by a glycerin, a different pattern. And cardiolipin, a membrane phospholipid in which two phosphatidic acid moieties share a single glycerol head group, and two phosphatidic acids share a single glycerol. R1 and R2 are fatty acid groups. It was first isolated from heart muscle and constitutes 15 to 20% of the total phospholipid phosphorus mass of the heart. It is found in all plants and animals. It is also found in bacterial membranes and the inner mitochondrial membrane of phospholipids, inner mitochondrial membrane of mammals. It is the only known antigenic phospholipid molecule. Cardiolipin is an important phospholipid in mitochondria and is involved in cellular energy production in the form of ATP. It is involved in the regulation of mitochondrial shape and function. And changes in cardiolipin levels can affect mitochondrial morphology and function. It also plays a role in apoptosis. Furthermore, this key phospholipid is known to play a role in disease processes such as heart failure, diabetes, thyroid hormone diseases, and genetic problems such as BARF syndrome. My dear friends, we reached the last glycerophospholipid, phosphatidylinositols. The phosphatidic acid in its structure contains an inositol group instead of nitrogenous bases. And these phospholipids act as intracellular signaling agents. Inositol is a polyol and sugar alcohol. It's an alcohol of glucose. And myoinositol, the myoconformer inositol, is included in the structure of this group of lipids. Phosphoinositides are composed of 1 mole of glycerin, 1 mole myoinositol, and 2 moles of fatty acid plus phosphoric acid, ranging from 1 to 3. So they may contain multiple moieties of phosphoric acid, and they are widely found in nature. Phosphatidylinositols are a minor component on the cytosolic side of cell membranes. However, their role is great. They are involved in lipid signaling, cell stimulation, and membrane trafficking. And here are the phosphatidylinositols. As you see, there are monophosphated ones, derivatives, 3-phosphate, 4,5, and D, 3,4-biphosphate, 3,5-biphosphate, and 4,5, and also 3-phosphate, 3,4,5,3-phosphate. So these phosphated derivatives act at various levels to regulate cell structure and metabolism. For example, phosphatidyl 4,5-biphosphate on the cytoplasmic phase of plasma membranes serve as a reservoir of messenger molecules released within the cell in response to extracellular signals that interact with specific surface receptors. And here you see the asymmetric distribution of phospholipids between the inner and outer monolayers of the erythrocyte's plasma membrane. And the distribution of a given phospholipid is determined by treating the intact cell with phospholipase C, which cannot reach lipids in the inner monolayer, but removes head groups of lipids in the outer monolayer. And the proportion of each head group released provides an estimate of the fraction of each lipid in the outer monolayer. And here you see the phospholipids we studied. Not all of them, sorry. This sphingomyelin is the part 3 topic, which is a sphingophospholipid. And you see the percentage in our inner and outer layer. Look at the choline and ethanolamine, for example. The ethanolamine rich in inner layer of the cell membrane, but choline is the outer, and also sphingosine here. And the rest almost reach in the inner monolayer or the inner layer of the cell membrane. And my dear friends, this is the end of lipids part 2. And here are the references I used to create this document for you. And also you can get one of them to study, to widen your knowledge. And the next topic is Lipids-III. We will study part 3. For more about me and the biochemistry, you can visit my website, biochemia.vet, and you can follow my Instagram account. And please don't forget to subscribe to the channel, to like the videos, and turn on notifications to be informed about up-to-date informations. See you in our next topic. Goodbye, my friends. Take care of yourselves and beloved ones. Ciao.